All right, so we're going to take a deeper dive into the molecular biology right now, and I'm going to show you how my lab tries to make a Google map of cancer and why it actually matters. All right, so first of all, I think it's really telling that one of these hallmark papers is the hallmarks of cancer paper. They changed their name and they recognize right now that a tumor is really complex and it should be considered a systemic disease. And so the way we try to look at that, we try to look at like not only the cancer cells, but also non-cancer cells, how they interact with each other, is through ideas about spatial data science. And spatial data science has been around for a very long time. Think about ecology, geology. And so we implement all these new high-tech technologies that identify individual cells. And then within these individual cells, we can identify individual transcripts. We don't only implement these ideas, but we also think about the software side. Like we can generate this really big data, but how can we get insights? And so we develop our own software, and that was now recently published or is accepted in principle in Nature Methods. All right, so just illustrating like how important and how much information we can get from these spatial omics technologies. I want to show this one slide. It's a core needle biopsy, two core needle biopsies from breast cancer patients. From one core needle biopsy, we were able to generate a really high quality single cell RNA-seq data sets. We got information from 5,000 cells. Yay, super amazing. Then from the same matching core needle biopsy, we were able to generate a 3D spatial proteomics, 3D spatial transcriptomics data set, a spatial imaging mass spectrometry data set, a spatial ATAC-seq data set, multiple H&E slides that we can use to annotate, and we can do many, many more. So for the same tissue input, we get information from multiple modalities and probably millions of cells compared to the 5,000 cells. So that was a single core needle biopsy, but can we do more? We can now easily generate data from multiple biopsies all at the same time within weeks, and we can unlock the potential of some of the biobanks that are available here on this side. So just as a quick example, here we wanted to see there's an Oncotype DX, which is a specific clinical assay in the clinic that they use to determine whether ER-positive breast cancer patients should receive additional chemo or not. It's a really simple 21-gene QRT-PCR test. And so there's a lot of flaws, and it actually has been shown that it doesn't work as well for Black women. So what we did is we took some of these ER-positive breast cancer samples and we applied our favorite spatial transcriptomics. And you can clearly see, and I'm just highlighting two of these biopsies, that they're very different. Every biopsy is very unique. Every patient is unique, not only in cell type composition, but also how these different cells kind of like are organized with each other. And if you then look at the right, that also illustrates like how some of these genes that you see in the y-axis, how much is being contributed by the different cells and how these different cells, depending on how they're organized, express more or less of these different genes. And so that contributes to that final score. And we're now trying to see what are the exact flaws and can we interrupt. And then this is just the last slide, just showing another really cool technology that is supported by the ALSF, the Crazy 8 Foundation, where we inject osteosarcoma cell lines within the tail of these mice. They have the propensity to go to the lung like they see in the patients. And these pediatric osteosarcoma patients, that's actually the main cause of death. And we see that, and if you look at this one specific lung lobe, there are multiple lesions, and all these lesions are super heterogeneous, where we see not only differences in gene expression, but also in cell-cell interaction. And so we're now thinking about, can we identify all the different vulnerabilities of these different specific clones or different specific subtypes, and how should we move forward with personalized medicine, not only in these models, but also in patients? Thank you.